Bombshell report today. We're going to be reading off of a Newsweek report that came out very recently that asked some very hard questions about where COVID-19 might have come from. And the questions are starting to really pile up around the idea that this was a creation of humans in some way, shape, or form. Hello, everyone. This is Dr. Chris Martinson here with your SARS-CoV-2 daily update, aka the honey badger virus. The honey badger virus is a very tricky foe, will escape almost any enclosure you put it into. Today is May 1st, 2020, day 99 of our reporting, almost up to the big magic round number triple digits. Let's go now to the numbers. Don't see any there, so let's go to the live website here. So we're looking at yesterday's cases here on worldometers.info/coronavirus. United States here with 1,095,000 total cases. Death count down from yesterday's high of about 2,800 here, but more cases. It was 28,000 yesterday, day before yesterday, and 30,000 yesterday. So still lots of cases racking up here. And as we dig down this list a little bit, we see that the UK really piling on a lot of cases here, 6,000 cases pouring in yesterday, also 674 very tragic deaths there as well. Germany starting to really tick up here with more deaths starting to get reported in now around here. Still a fraction. Look at this. Germany compared to France, 163,000 identified cases here for Germany, 167,000 for France, but 6,600 deaths compared to 24,376 deaths there. As well as we go down the list, we start to see Brazil having a very hard time of it here. Lots and lots of deaths showing up in Brazil. China, yeah, I'm just going to laugh at these numbers. Yeah, right, +4 with a major city shut down. You know how China is. They're going to shut down a major city because 4 new cases. Not even close. So again, China, keep trying. Maybe you can join the world's numbers parade at some point, but not with... You make it 6 next time. More believable. All right, Canada starting to really show up here as well. 1,600 more cases showing up, 188 deaths there. But what I wanted to really talk about is down here in Peru, moving into their fall down there. So much more theoretically conducive environment for the spread of this particular virus. 3,000 new cases, 108 deaths there in Peru yesterday. Switzerland seeming to has got its arms around what's going on there. Remember, they used to be up at the top of the list in terms of daily counts, thousands and thousands of cases. But Saudi Arabia suddenly having 1,300 cases now. They used to be handling it that a lot better, I would think, and a lot lower numbers. Mexico also really starting to show up. I think poor reporting is partly to explain even why this number is as low as it is. And Singapore back having some trouble with some cluster outbreaks. Case, cluster. They're having a few clusters, so they're fighting those. And so anyway, they're having their difficulties. Chile, next to Peru, having their difficulties here. And interestingly as well, both Qatar and the UAE, both of which had done very, very good jobs containing things up to this point, also beginning to struggle. So the honey badger virus, soon as you let things off for just a little bit, this is what you get. The cases come roaring back out again. So I think that's what we're going to see in the United States with the relaxation and the opening up of social distancing and opening up businesses and things like that. We'll see more cases come out again, and then we'll have to decide how we're going to handle it, just like every country has to make that same decision. How open, how closed, how many cases, how many deaths are you willing to entertain? South Korea, though, still very much, these are believable numbers because they do lots and lots of testing. And again, if any of these countries, including mine, the United States, wanted to know what to do, you could do worse than to go over and take a little fact-finding trip to South Korea and see how they do what they do. Very, very elegant systems of contact tracing, use of technology to help people understand where the cases are, all of that. So at any rate, all over the world, you just keep scrolling down. It's pretty much everywhere. And interesting here, Diamond Princess, still, this column here is active cases. 54 active cases, 4 still in serious or critical condition. Remember, no more than that original 712. This first broke on February 4th. February, March, April, we're closing in on 3 months, 90 days later, and there are still active cases, and there are still people in serious critical condition. That's just horrifying. So again, this ain't the flu. All right, we're going to turn now to a clip from the movie Jurassic Park. Let's listen in.
I'll tell you the problem with the scientific power that you're using here. It didn't require any discipline to attain it. You read what others had done, and you took the next step. You didn't earn the knowledge for yourselves, so you don't take any responsibility for it. You stood on the shoulders of geniuses to accomplish something as fast as you could, and before you even knew what you had, you patented it and packaged it and slapped it on a plastic lunchbox, and now you're selling it. You want to sell it. Well,
I don't think you're giving us our due credit. Our scientists have done things which nobody's ever done before.
Yeah, but your scientists were so preoccupied with whether or not they didn't stop to think if they should.
All right, I raised that because I want to bring you to this bombshell report that came out in Newsweek. And so this came out a number of days ago. I've been digesting it. So what's that, 3 days ago? This is really important information. I haven't really seen it picked up in a lot of other places, but this is really a bombshell, and it deserves our highest attention. It really does. And I've been sitting on this information for a while. I've had the information that was in this article. They've given me some new stuff as well. Very well-written articles. So I'm not trying to say I scooped them, but I've been sitting on it because I didn't know what to do with this information. I didn't want to come off like a nutter or didn't want anybody to think I was reaching too far. But now that it's in Newsweek, I think that helps. And so I can talk about this more openly. Dr. Fauci, remember who Brad Pitt has played and then took his wig and makeup off and thanked Dr. Fauci from the bottom of his heart for doing what he does. But this same Dr. Fauci backed controversial Wuhan lab with millions of dollars, US dollars, for risky coronavirus research. Let's check in here. Dr. Anthony Fauci is an advisor to President Donald Trump and something of an American folk hero for his steady, calm leadership during the pandemic crisis. At least one poll shows that Americans trust Fauci more than Trump on the coronavirus pandemic, and few scientists are portrayed on TV by Brad Pitt. But just last year, the National Institute for Allergy and Infectious Diseases, the organization led by Dr. Fauci, funded scientists at the Wuhan Institute of Virology and other institutions for work on gain-of-function research on bat coronaviruses. I'm going to tell you what gain-of-function is in a second. Really important phrase there. We'll get to that in just a second. But first, we'll note here, in 2019, with the backing of the NIAID, the National Institutes of Health committed $3.7 million over 6 years for research that included some gain-of-function work. The program followed another $3.7 million 5-year project for collecting and studying bat coronaviruses, which ended in 2019, bringing the total to $7.4 million. So the first thing they did, they spent 5 years collecting and studying bat coronaviruses. And then, starting in 2019, started monkeying around with gain-of-function work on those collected viruses. So they went out into nature, went into the caves, collected all these viruses, probably amplified them, studied them, did some stuff, but then they went went into gain of function. So what is this gain of function? This, also from the NIH, talks about certain gain of function studies with the potential to enhance the pathogenicity or transmissibility, or both, of potential pandemic pathogens, PPPs, have raised biosafety and biosecurity concerns, including the potential dual-use risks associated with the misuse of the information or products resulting from such research. So you remember that clip we just watched? Just because you can do something doesn't mean you should do something. Well, guess what? In October 2014, the White House Office of Science and Technology Policy announced the launch of the U.S. government gain-of-function, the GOF, gain-of-function deliberative process to reevaluate the potential risks and benefits associated with certain GOF, gain-of-function experiments. So in 2014, the government said, hang on, wait a minute. This is a little scary even for us. Just because we can do this doesn't mean we should do this, right? So during this process, the United States government paused, didn't make it illegal. They paused the release of federal funding for gain-of-function studies anticipated to enhance the pathogenicity and/or, they should put and/or in there, not just or, transmissibility among mammals by respiratory droplets of influenza, MERS, or SARS viruses. Okay, so then they studied that for a while, and then they thought about it. But then in January of 2017, the United States government released policy guidance for review and oversight of research anticipated to create, transfer, or use enhanced PPPs. And guess what? They kind of said, you know what? Yeah, we think we can control this. We think we could do it safely. Let's open up research back on gain of function on bat coronaviruses, among other things.
But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
Yeah, just because you can do something doesn't mean you should do something, right? So let's, this is the second, these little numbers here are /2. You'll see them up to about /6 in this report. Tells you that this is the second quote that I'm pulling from this one Newsweek article, okay? SARS-CoV-2, the virus now causing a global pandemic, is believed to have originated in bats. In fact, its bat sequence homology is about 96%, I believe, or I think that's right. Well, it's very high. I've read different reports about which bat coronavirus it's closest to. I think there's one coronavirus it's 96% homologous to. I think it's about 85 or 88% homologous to actual SARS itself. But even if you said, oh, it's 96%, it must have come from a bat. Remember, humans share something like 98% genetic homology with bonobos, our closest primate relative. So even a percent or two is a big deal. Makes a big difference in these sorts of things. So 96% homologous doesn't mean it's the same as. It could be very different. U.S. intelligence, after originally asserting that the coronavirus had occurred naturally, conceded last month that the pandemic may have originated a leak from the Wuhan lab. At this point, most scientists say it's possible, but not likely, that the pandemic virus was engineered or manipulated. And we're going to talk about which scientists say that. And by the way, U.S. intelligence holds no value for me anymore. Like, who? What do you mean U.S. intelligence? Who is that? Is that the CIA? Is that the NSA? I mean, that's a little vague for me, so I don't know what that means, U.S. intelligence. And don't forget, U.S. intelligence missed the collapse of the Soviet Union and also thought there were weapons of mass destruction in Iraq. So U.S. intelligence doesn't always get things right. That's all I'm saying here. Okay. Dr. Fauci did not respond to Newsweek's request for comment. NIH responded with a statement that said, in part, most emerging human viruses come from wildlife, and these represent a significant threat to public health and biosecurity in the U.S. and globally, as demonstrated by the SARS epidemic of 02-03 and the current COVID pandemic. Scientific research indicates there is no evidence that suggests the virus was created in a laboratory. This is from the NIH. The key word here is most. Emerging human viruses come from wildlife. Most. They didn't say all. It's an important little qualifier. That's a weasel word right there. Most. And the one in question, of course, is this one. Scientific research indicates there's no evidence. Oh, really? What kind of research? Well, let's take a quick peek at that, shall we? Yes, we will. All right. So let's move on here. Let's look into that research. Here's some research that came out. This came out in Nature in March 17th, 2020, the proximal origin of SARS-CoV-2. So proximal means near. So where'd this come from? By the way, some people in here, Ian Lipkin, he was one of the main consultants for the movie Contagion. That's why you might have seen that name before, but it bears noting that these people here all do a lot of research on the kinds of things here that we're talking about. So just always keep that in mind. Because remember, you show me the incentive, I'll show you the conclusion or the outcome. All right, there's a whole big long paper up here, and there's a lot of science above it. But here's where they got, this is the money shot in this particular paper. They said, while the analyses above suggest that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict the interaction is not ideal and that the receptor binding domain sequence is different from those shown in SARS-CoV itself, classic SARS, to be optimal for receptor binding. Thus, the high affinity binding of SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection on a human or human-like ACE2. Okay, all right. This is really bad logic. Let's break this down, because here's what they just said. Computational analyses predict that the interaction is not ideal, but actually it is a highly ideal binding arrangement. So what they're saying is because our computers and our models that we're using couldn't have predicted that the sequence that we see is actually going to be a really good sequence. Therefore, it couldn't have been human-derived because our computers wouldn't have spit out that sequence, so we never would have tried that sequence. However, that's one way you can go about trying to create a virus. One way is you can try and create a computational sequence, and then you make that sequence, you put it in the virus, you test it. There's another way you can do this too, which is you can yank other sequences known to work, and you can insert them into parts. And you can also do gene editing, and you can do random mutations, and you can do other things where you can use the power of the laboratory to create all these random mutations, test them against cell lines and against binding assays, and then find one that seems to really work, and then use that sequence and amplify that and then put that back in. All they're saying here, which was a little weaselly, a little mealy-mouthed, they're saying because a computational analysis predict this is not an ideal binding, but it actually is. They're saying thus, the high affinity binding of SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection. So yeah, that's not how any of this works. Because you can't, that's one conclusion. There's a second conclusion, which is, is there another way? Is there another way that we could go about, monkeying with these genes rather than computing them? And there is. Of course there is. And so this is really faulty logic. I'm kind of surprised that made it into Nature. I really am. Because usually the stuff is more well peer reviewed than that. But again, if the other people peer reviewing it happen to be other people who are conflicted about this whole thing and really wouldn't like it to be revealed that virologists and monkeying with viruses is a bad idea, and their paycheck depends on that not becoming a well-known fact, then I could see this sneaking through. But this is faulty logic to say because a computer didn't predict this or wouldn't have predicted this, therefore it didn't come from humans. All right, I'm going to show you why in just a second. So remember, we had this NPR piece on where they had all these experts. 10 experts all agreed that this had to have come from a natural source. Couldn't have come from a lab, right? It was super weak refuting. I tore that apart a few days ago. This is another piece we didn't really go into. I want to just show you that there's a meme out there, and it looks like this. Regardless, genetic analysis shows the virus began to spread sometime in the fall or winter of 2019, says Robert Gary, a microbiologist at Tulane University. Those same analyses refuted an earlier theory that the virus was genetically engineered in a laboratory. Gary says the reason is simple. The virus infects people in a way that scientists had never seen before. The virus is just really good at what it's doing. He says no human being using a computer could do this. This is very clearly a natural process that occurred. In addition, he says there are no signs of genetic modification in the virus's code. Robert Gary, wait a minute, didn't we? Oh yeah, there he is, right there, saying the same argument again out here, which is because a computer couldn't have predicted it and no human using a computer could do this, it's very clearly a natural process that occurred. Well, that's interesting. Well, let's look into something real quick. This has been around a long time. This is infectious DNA cloning. Look, 2009, this article came out. But even more than that, the development of recombinant DNA methods by Cohen and Boyer in 1973, together with the discovery of reverse transcriptase by Temin in Baltimore in 1970, made it possible to introduce a mutation at any location in a viral genome. The essential reagent is an infectious DNA clone, a double-stranded DNA copy of the viral genome carried in a bacterial plasmid. These DNAs, or RNAs produced from them, can be introduced into cells by transfection to produce infected viruses. Continuing on, the infectious viral DNA clone is a double-edged sword. It enables the manipulation of the viral genome at will, allowing unprecedented genetic analysis and the use of viruses as vectors for gene therapy. That could be a good use. But nearly any virus can now be recovered from the nucleotide sequence, effectively making it impossible to ever truly eradicate a virus from the globe. So this has been around now, just to be clear, since the 70s. It's only gotten better and better. So to say that the only way that you could really come up with a new virus, the only way that you could really monkey with a virus is using a computer, is not actually true. You can also do it by the brute force method of just putting random mutations in, or you can be smarter about it, and you can start inserting cassettes from other viruses into other parts of the viral genome. That's all pretty standard work at this point in time. All right. So, continuing on with this super weak refutation, here, Jonah Mazet, that's who the Mazet is here, says, lowering the odds further still, when researchers begin to work in the lab to see what they've collected, the samples they handle aren't actually infectious. Mazet says they are inactivated, a chemical process that breaks apart the virus itself while preserving its genetic material for study. So it's inactivated. It's just chunks, just totally ruined chunks, no longer capable of being infectious. We basically deactivate or kill the virus before we work with it to limit the possibility of accidents, she says. And then we work inside biosafety cabinets even on top of that. So there you go. They're just dead little fragments. They're just dead little fragments that we're working on. And no human computer could do this. This is the story that is being run out there right now. And by the way, all these people, it's a very small world. These people, Jonna has worked with Gary, who's worked with Peter, who's worked with all these people. They all work together. All right. So let's carry on then. The NIH research consisted of two parts. The first part began in 2014 and involved surveillance of bat coronavirus that had a budget of $3.7 million. The program funded Shi Zhenling, the bat lady, the bat woman, a virologist at the Wuhan lab, and other researchers to investigate and catalog bat coronaviruses in the wild. This part of the project was completed in 2019. A second phase of the project beginning that year included additional surveillance work, but also gain-of-function research for the purpose of understanding how bat coronaviruses could mutate to attack humans. The project was run by EcoHealth Alliance, a nonprofit research group under the direction of President Peter Daszak, an expert on disease ecology. NIH canceled the project just this past Friday on April 24th. Bing! Too little, too late, I think. Politico reported Daszak did not respond immediately to Newsweek for requests for comments. Gain of function. We're doing gain of function stuff on here. So remember, we talked about this a number of days ago. I raised this exact same thing here because look at this. Remember, you said here, despite the evidence, misinformation about the virus's origins continue to proliferate, said Peter Daszak. For Daszak, who has worked on other outbreaks, the pattern is all too familiar. Every time we get a new virus emerging, we have people that say, this could have come from a lab, he says. What's unusual about this outbreak, he says, is that senior officials in both China and the U.S. have traded accusations that each nation is somehow responsible for the virus. It's a real shame that the conspiracy theories can get to the level they've got with policymakers. That's it. As soon as you declare something a conspiracy theory, you've thrown shade at it. You've thrown mud at it, right? Conspiracy theories. It must be wrong, right? It's such a weak dodge. It's intellectually bankrupt, honestly. The political heat has strained the very scientific collaborations meant to detect those viruses as they emerge, warns Jonna Mazet here. There's that name again. The PREDICT system she runs relies on the voluntary cooperation of many countries, with researchers sharing data about viruses they're spotting within their borders. With this sort of blame game that's going on, we put ourselves at much more risk. Daszak says the time for finger pointing is over. We're done. Too much finger pointing already. Too much. Why? Because we have a bat virus in my neighborhood in New York and it's killing people, he says. Let's get real about this. So I got a little more curious because Daszak, his name keeps showing up, right? And this, this, all the work that he's been doing. So guess what? Power of the internet. Let's look at the gain of function research. So here's a big thing. This is really tiny. I'm not going to try and read this. I'm going to pull out for you. I've copied it directly. This language right here. We're just going to take a quick peek at this, okay? Because this talks about understanding the risk of bat coronavirus emergence. This is Peter Daszak right here. It's a funded project. And when did it start? Well, let's take a quick peek. Project start date, June 2014. The budget start date is July of 2019. And project end date, 24th of April. $292,161. Let's look at that aim number 3 right here again, but up close. Aim 3, what was the aim of Daszak's research? It was in vitro and in vivo characterization of SARS-CoV spillover risk. And spillover risk, by the way, to decode that, is the risk of something spilling over from one species to another, for it to actually come out of bats, to end up in some other species, maybe pangolins, maybe snakes, maybe ferrets in the lab, and then into humans. Okay, so that's spillover risk. Coupled with spatial and phylogenetic analyses to identify the regions and viruses of public health concern, we will use, this is the money shot right here, S protein sequence data. That the S protein is the spike protein that does the binding. That's where the receptor binding domain is. Infectious clone technology, which we just talked about up here. What is infectious clone technology? That's where you can pretty much put any mutation into any virus you want at any place you want. So they're going to be messing with the DNA, the RNA sequence of this particular virus. Why? Because they want to conduct in vitro. That's in glass. So that would be various experiments that you would see in vitro, and in vivo infection experiments. In vivo is in animals. That's in live animals. So just to decode this one thing, they're going to take the binding domain, they're going to put mutations into it, and then they're going to look in vitro and in animals to see if they can gain a function, if they can improve its ability to be either transmissive or more pathological. That's what this science is about. And to continue our analysis of receptor binding to test the hypothesis that percent divergence thresholds in S protein sequences predict spillover potential. So they are actively monkeying with the receptor binding domain to ask the question, well, if we had changed a couple of amino acids, or if it had one mutation, if it had two mutations, well, if it had 4 or 5 mutations, the question they're asking, which I think is a decent question to ask. It's a question you can ask. Should you ask that question? It's a different question. They are asking the question how much of a mutation would be required for this thing to jump out of bats and come into humans. And they were running those exact experiments. And those experiments were being run in Wuhan. And the next thing you know, we have this thing breaking out of Wuhan. Remember.
But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
All right. This is going to number 4 here. According to Richard Ebright, an infectious disease expert at Rutgers University, the project description refers to experiments that would enhance the ability of bat coronavirus to infect human cells and laboratory animals using techniques of genetic engineering. That's what all of this stuff was. That's what this junk was. That was the stuff that Fauci had funded out of the NIH and was funded to this gentleman, Daszak, who was running these exact experiments, who, when given the chance, told NPR, it's just crazy, wacky conspiracy theories, just a blame game. This isn't time for finger pointing, and there's no chance that this could have come from anything but a natural reservoir, just bad luck. Here's the understatement of the year award. In the wake of the pandemic, that's a noteworthy detail. Yeah, I would say that is. Let's read it again, because in the wake of the pandemic, this may well be a noteworthy detail. The project description refers to experiments that would enhance the ability of bat coronavirus to infect human cells and laboratory animals using techniques of genetic engineering. That is a very noteworthy detail. How has that been skipped over in all of the research reports that are out there, including this one in Nature, which should have been much more well peer-reviewed, and this whole junk idea that only computational analyses could have done this, and since it didn't predict this sequence, or might not have, therefore it had to have been a natural thing, is absolutely faulty logic of the highest order. Ebright, along with many other scientists, including me, raising my hand here, raising both hands, got both my hands in the air, has been a vocal opponent of gain-of-function research because of the risk it presents of creating a pandemic through accidental release from the lab. So this is what you're supposed to be doing as scientists. They convened that panel when in 2017 they released an 1,100-page report saying, basically, let me summarize all 1,100 pages. You know what? I think we can control the risk of this, and because we can control the risk of it, we would like to do these experiments, because you know what? We all earn a nice paycheck from these experiments. They're kind of interesting, and it's kind of fascinating work, and we think we can do this work, so we should do this work. And that's really, Ebright, I totally agree with you here. All right. Here it is. Check this out. A decade ago, during a controversy over gain-of-function research on bird flu viruses, which was an awful thing. Somebody created a bird flu virus in the lab that had the capability of that bound to human cells in a laboratory, and also infected ferrets, which is a useful model. That was awful. If that stuff had ever gotten out, or somebody got their hands on the sequence and decided to make it in some lab as a bioterrorism thing, that's awful. So at any rate, there was a lot of controversy over that. And Dr. Fauci played an important role in promoting that work. He argued that the research was worth the risk it entailed because it enables scientists to make preparations, such as investigating possible antiviral medications that could be useful if and when a pandemic occurred. Of course, it wouldn't be useful if you accidentally made a pandemic virus that escaped before you could also make useful antivirals. Remember. All right. Oops. Hold on one second. Gonna get this center back up again. There we go. Back on it. The work in question was a type of gain-of-function research that involved taking wild viruses and passing them through live animals until they mutate into a form that could pose a pandemic threat. Scientists used it to take a virus that was poorly transmitted among humans and make it into one that was highly transmissible, a hallmark of a pandemic virus. This work was done by infecting a series of ferrets, allowing the virus to mutate until a ferret that hadn't been deliberately infected contracted the disease. The work entailed risks that worried even seasoned researchers. More than 200 scientists called for the work to be halted. The problem, they said, is that it increased the likelihood that a pandemic would occur through a laboratory accident. And guess what? We have laboratory accidents all the time. Many, many things have escaped from laboratories. So if we're out here in using bad viruses, collecting ones that are really wicked close to the ones that could infect humans, and then doing genetic experiments on them to create gain-of-function experiments, tweaking them in vitro so you get a really good sequence that binds really well, then doing your in vivo experimentations on ferrets or maybe other lab animals, and the next thing you know, you've got this thing and it's awesome. It infects and transmits, and it's quite transmissive, and also maybe just a little bit lethal. That was the goal of the study. And then what happened next? Well, I think we all know what happened next here at this point in time. Dr. Fauci defended that work, determining the molecular Achilles heel of these viruses can allow scientists to identify novel antiviral drug targets that could be used to prevent infection in those at risk, or to better treat those who become infected. He wrote back in 2011 in the Washington Post, decades of experience, he wrote, listen to this voice of authority, decades of experience tells us that disseminating information gained through biomedical research to legitimate scientists and health officials provides a critical foundation for generating appropriate countermeasures and ultimately protecting the public health. That sounds reasonable, right? But look at this. Disseminating information gained through biomedical research. Dude, this isn't biomedical research. This is tinkering with a virus. This is tinkering with something that's, if it gets away from you, the downside of that is a collapsed world economy in a pandemic. The upside is, we learned a few things that were kind of fun and interesting, and all my lab mates were, and the colleagues across my profession were kind of impressed with me. That upside is minuscule compared to the downside, but viral research is not disseminating information gained through biomedical research, not when you're doing live animal gain-of-function testing. Get out of here. Get out of here. That's dissembling. That's what that is right there. All right. Continuing on then. Nevertheless, in 2014, under pressure from the Obama administration, the National Institutes of Health instituted a moratorium on the work, suspending 21 studies. 3 years later, though, in December 2017, the NIH ended the moratorium, and the second phase of the NIAID project, which included the gain-of-function research, began. The NIH established a framework for determining how the research would go forward. Scientists have to get approval from a panel of experts who would decide whether the risks were justified. Ooh, I bet we're all really happy with that now, huh? The reviews were indeed conducted, but in secret, for which the NIH has drawn criticism. The reviews were conducted, but in secret. The reviews were indeed conducted, but in secret. But in secret? Why would you hold those in secret? Yeah, I guess you could say there's a fair bit of criticism that's determined there. That's useful. In early 2019, after a reporter for Science magazine discovered that the NIH had approved 2 influenza research projects that use gain-of-function methods, scientists who opposed this kind of research excoriated the NIH in an editorial in the Washington Post. So this isn't like all scientists kind of agree on this. A huge body of scientists say it's a bad idea to do gain-of-function research because these things escape from labs all the time. That's the nature of them. They're really, really hard. That's why you have these incredibly ridiculous, the BSL-4 lab, you got the people in the moon suits and you got the positive pressure suits and you have all these decontamination rooms and there's airlocks. You know why we do that stuff? Because these little virus particles are very, very difficult to contain, particularly when you're doing live animal studies. So you got ferrets, they're in cages. Somebody has to go in and feed them. You got to change the litter. It's just daily stuff you got to do. Maybe they're handling the ferrets and then you got to inject them or measure them or take temperatures or whatever you're doing, right? There's all the experimentation, drawing blood. Every one of those steps, there's a possibility that a virus particle or particles can somehow escape, get into the suit, remain on the suit in a fold. Listen, it's just, there's a chance. There's always a chance. So given that there's a chance for something to go wrong, a lot of scientists said maybe we shouldn't be doing this stuff. All right? Just because you can do something doesn't mean you should. All right. Yeah. Here's my summary of all this. In partnership with the Wuhan Virology Institute, the U.S. NIH was funding the gathering of and gain-of-function experimentation on bat SARS viruses of the exact genotype of the one that is now parading across the world, creating a capital D depression and ruining millions, if not billions of lives and costing untold hundreds of thousands of deaths. The experimentation included genetic alteration of and in vivo live animal testing of these SARS bat viruses and their genetic altered variants. In late 2019, COVID-19 or SARS-CoV-2 erupted within just a few hundred yards of the Wuhan Virology Institute where all this work was being conducted. And then all the scientists, not all of them, but most of them directly involved with this research, they've all been on a public relations spree to explain that the virus is 100% natural and couldn't possibly have come from a lab. However, we need to consider that, exactly the sorts of genetic manipulations that could have led to SARS-2 were being done, right? And B, even if they weren't, this still wouldn't refute the idea that a wild-type virus was gathered, it was amplified, and accidentally released from a lab. So there hasn't been if these scientists who want to say there's no chance this came from a lab, if they wanted to actually maybe win me over a little bit and maybe go, oh, okay, they would soften that and they would say, you know what? We can't say for sure whether it came from a lab or not. It seems unlikely to us because of X, Y, and Z. You know what? Somebody could have gathered this, amplified it, and then it could have gotten away from them in a lab, but we don't think so because X, Y, and Z. But they're just saying flat out, definitively, declaratively, oh no, it's wild, didn't come from a lab, right? That self-serving sort of statement coming from these particular individuals falls really flat on my ears. In fact, worse than that. It starts to sound highly suspicious to me. Conclusions for today. The press needs to start asking Fauci and everybody else involved some really tough questions. And we ought to ask ourselves what the penalties ought to be if it turns out that this was something that was just a really terrible set of decisions to have been done, and if this was some sort of an accidental release of some kind, and something worse and darker than that. All right. The string of coincidences around the fact that the Wuhan Virology Institute was exploring gain of function of precisely the virus now infecting the world is very long, a very long string of coincidences, right? Oh, we just happen to have been doing research on those exact viruses, and we were doing exactly the sort of genetic manipulations and the exact sort of S protein receptor binding domain genetic manipulations and mutations, coupled to in vivo work that could have accidentally led to something like this, but trust us, that's not what happened. These definitive statements by the involved scientists that it couldn't have been man-made and it didn't come from a lab, they're not factual statements, right? That was that sophistry that, our computational models wouldn't have predicted this exact sequence, therefore it wasn't humans. That's just not how any of this works, right? Because there's another way you could do this that doesn't involve computationally deriving it. You can do it the good old-fashioned way, inserting point mutations in a critical zone, or going to other viruses that have that exact binding domain and yanking those and cassetting those into the receptor binding domain to see what happens. Listen, even with all that, this could have been naturally gathered, amplified, and released, or it could have been gene edited, not computationally derived, right? There's all these things that we can know that at any rate, these things are possibilities. And so we have to hold open that possibility. But the sequence of things here is very, very suspicious. And I hope we get to the bottom of this. What's the most important thing I can tell you? Regardless of whether any of this is true, regardless of whether this came from the Wuhan lab or not, regardless of any of that, we should not be doing these experiments. Full stop. And anybody who says we should be doing them is a danger to society, is a menace, because the upside to this thing is tiny. The downside is absolutely massive. And so that's the full stop. No questions about it. That's where I land on this whole thing. Just because, remember. All right. With that, I'm all done for now. Hey, have a great weekend. Plant a garden. That's what I'm going to be doing. All of my garden starts are up and out. I think this virus is going to be with us for a very long time. I think it's a beast. It's a honey badger. I think we're not going to be getting rid of it anytime soon, and we're just going to have to learn to live with it. And regardless, there's going to be some downstream economic impacts from this that are going to be really just absolutely far more massive than anything we've seen all up to this point in time. So with that, thank you very much for listening. Have a great weekend, and we'll be back with you on Monday.
Hi, folks. Adam Taggart, co-founder of Peak Prosperity, here with good news. In his daily videos, Chris often refers to the book that he and I wrote, Prosper: How to Prepare for the Future and Create a World Worth Inheriting. It presents a practical model for living with greater resilience, and those who read it and followed its advice were much better prepared for the coronavirus outbreak than the average household. Given its heightened relevance to the turbulent times we now find ourselves in, Chris and I want everyone to benefit from this book's formula for better, more secure living. So we're giving the book to you for free. To get your copy, simply visit peakprosperity.com/freebook. We think you'll find this an extremely valuable and inspiring resource for the current COVID-19 outbreak and for being well positioned for any kind of crisis that comes our way in the future. Thanks for listening, and happy reading.